Hello everyone and welcome to another recorded lecture of BIO1 Online. Today we'll discuss Chapter 8, which is on DNA replication and mitosis. Most of this conversation will be on the cell cycle, which includes mitosis and cell division, but let's start talking about DNA replication first. DNA replication precedes cell division. In cell division, a parental cell divides into two daughter cells. So we call the original cell the parental cell, or the parent. And as that cell divides, it divides into two daughter cells. And for each of the daughter cells from the division to have identical DNA, the parent cell must first replicate its genome. Right, this makes sense. If a parent cell just divided into two, then each of the daughter cells would have half of the genetic information as the parent. We want the parent cell to replicate into two identical cells. So we first need to replicate all of the genetic material in the parent, and then we can divide them into two. I would like to watch a quick video that overviews DNA replication first, before we talk about the details.
DNA replication is the process in which one double-stranded DNA molecule is used to create two double-stranded molecules with identical DNA sequences. For this process to occur, the two DNA strands are separated, and the DNA replication machinery is assembled at the replication fork. The two DNA strands are used as template strands to create newly synthesized DNA strands. The key feature that allows a DNA molecule to produce two DNA molecules with the same DNA sequence is the ATGC rule, also known as Chargaff's rule. According to this rule, adenine in one strand only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. For example, an incoming nucleotide with a cytosine base will only hydrogen bond with a guanine base in the template strand. By this mechanism, one double-stranded DNA molecule provides the information to create two double-stranded molecules with identical base sequences. Each copy contains one newly synthesized strand and one original strand. This mechanism is called semiconservative DNA replication because half of the DNA molecule is conserved from the original molecule.
So hopefully that gave you a brief overview of what we're going to be talking about in more detail. Before we talk about eukaryotic DNA replication, let's really quickly go over prokaryotic DNA replication. It's pretty short and simple. Bacteria each have a circular chromosome, a single chromosome of DNA, and the process begins at a specific location called the origin of replication. At that origin, replication moves in both directions. So a certain enzyme called DNA polymerase, as we'll soon be very acquainted with, copies the circular DNA, goes in one direction, copies. Another one goes in the opposite direction until you get two identical pieces of circular DNA. So it's pretty simple. And replication is very quick. Replication takes about 40 minutes, and the cell divides every 20 minutes. So this means that every cell is in the process of replicating its DNA at some point, every bacterial cell. So these have to constantly replicate their DNA because bacteria divide by binary fission. And that's how they don't do mitosis. Bacteria divide by binary fission. So now let's take a little trip to eukaryotic land. Let's forget about bacteria for a second. When we're talking about eukaryotes, we have many linear chromosomes. Remember, we have 46 chromosomes in all of our cells. All 46 of these chromosomes have to be copied at the same time. So specifically, we have 46 double helices, right, wrapped around histone proteins, and that's what forms a chromosome. The first step in DNA replication is that the strands, the double helices, have to separate, that the unzip. The enzyme that does that is called DNA helicase. So DNA helicase is an enzyme that can break the hydrogen bonds between the DNA double helix. And that happens at numerous points along each chromosome. So there are these multiple locations called origins of replication. where helicase can start unzipping DNA by breaking those hydrogen bonds that keep the two strands together. Right? These hydrogen bonds are crucial. Once the two strands are separated from each other, each strand can be used as a template for a new strand. So basically, if one strand has an A, what will DNA polymerase want to put in? Well, we know it has to be a T because of the base pairing rules. So new complementary nucleotides are added in by the enzyme DNA polymerase. And at the end of DNA replication, we're going to have two identical DNA double helix molecules, right? Two identical chromosomes. Since half is newly formed and half is conserved from the original molecule, right? The original double helix and the parental cell was blue. But we use gold nucleotides to build the new strand. So since half is conserved, we say that DNA replication is semi-conservative, and that's a very important fact about DNA replication, right? This is a new DNA molecule, and this is a new DNA molecule. They resemble the exact same code as the parent, except these have new nucleotides incorporated, so this is semi-conserved. So again, step one is the strands have to separate by DNA helicase. Each strand serves as a template that attracts and binds complementary nucleotides. So if there's a C, then a G will be put in. If there's a T, then an A will be put in. And at the end, each double-stranded DNA molecule will consist of one parental and one daughter strand as a result of semi-conservative replication. This is another way to look at it. And where the two strands separate is called the replication fork. Kind of creates a fork in the road. Right? And that's where, at the replication fork, DNA helicase continues to unzip. So it's going to unzip the double helix going up this way. And as there's more space, as there's an exposed G, then a new cytosine can come in. If there's a G, another cytosine. A, then a T will come in. C with G. This is another way to look at it in 2 dimensions. So here is our original parent molecule. Each of these two strands separated. So here's one and here's the other. And then DNA polymerase started and copied in with complementary base pairs. All right, so if there is a C, then of course there's a G. Since there is a G on this template strand, it's going to put in a C. And what do you know? This is the exact same thing as this and this, right? From this one molecule of DNA, we got two identical DNA molecules. And all 46 chromosomes get replicated at the same time. So pause here and try to answer this rapid response question. Which of the following best represents the newly created DNA strands in red after DNA replication is complete? So the answer is C. Right, DNA replication is semi-conservative. And we said DNA polymerase is the enzyme that makes DNA. It does so by attaching complementary DNA nucleotides together according to whatever the template DNA sequence is. However, there's a catch. DNA polymerase can only add nucleotides to the 3' end of another nucleotide. So it has one direction it can go in. What this means is that DNA is only made from 5' to 3'. New DNA is synthesized 5' to 3'. I'm going to repeat this many times during this talk. So DNA polymerase can only make DNA 5' to 3'. So that means here's DNA polymerase. What end must this be? If it's adding to this end, this must be the 3' end. So DNA polymerase is making new DNA 5' to 3'. DNA polymerase recognizes the 3' end of the template. I'm going to talk a lot more about this too, but right now I want you to understand that DNA polymerase is made from 5' to 3'. It can only go in one direction. It can only add nucleotides to an existing 3' end. Therefore, it only knows how to make DNA 5' to 3'. It can only add to this direction. It can only add over here. It can't add anything in this direction. It can only add something to the 3' end. Therefore, DNA replication, DNA is being made 5' to 3'. right? Because things are only growing at the 3' end. And in this case, this would have to be the 3' end, and this would have to be the 5' end. Because remember, DNA strands are always anti-parallel to each other. If this is 3', that has to be 5', right? You can never have two 5' or 3' parallel to each other. So like I said, new DNA is made 5' to 3'. So with that said, at which ends of the template does DNA polymerase start reading from? So if DNA polymerase can only make new DNA from 5' to 3', it has to start toward the 3' end of the template. Right, so if here's DNA polymerase, right, since DNA is made anti-parallel, DNA polymerase must start replication at the 3' end of the template. And this makes sense, because DNA polymerase only knows how to make DNA 5' to 3'. It can't start here, because then you're going to have a 5' and a 5' next to each other, and we can't have that happen. So DNA polymerase cannot start at the 5' end of the template. This is the template strand. It has to start toward the 3' end of the template, because then it can make DNA 5' to 3'. This is the 5' end. What will DNA polymerase put here? If there is a T in the template, what will be put here? Well, an A, right? If there's a G, then what will be put here? C, G, and then T, and then 3'. That new DNA was made 5' to 3' because it started reading from the 3' end of this template strand. So there we have our code ACGT. So if you just look at the blue, was our parent molecule, we had two options on where to start. We could not make 5' to 3' DNA here, right? Because then we'll have two 5' on top of each other. That's not good. We were able, though, to make 5' to 3' DNA starting at the 3' end of the template. What enzyme polymerizes nucleotides? Pause here. And the answer is C, DNA polymerase. In which direction does it add new nucleotides? Pause here. The answer is A. It can only make DNA in 5' to 3' form. Where on the template strand does DNA polymerase start replication? So pause here. And the answer is B. Toward the 3' end is where it starts. So now let's break this down step by step. We said DNA helicase is the first member of the party who needs to replicate our DNA. So DNA helicase comes in and it can unzip the DNA double helix. It does so by breaking all the the hydrogen bonds between the two strands of DNA. So DNA helicase breaks hydrogen bonds and unzips genes. Once those two DNA strands are separated, they're likely to come back together since they're attracted to each other. So we need these SSB proteins, or single strand binding proteins, to kind of act like clamps. They clamp on each single strand, they bind to each single strand, preventing them from coming back together. So now we open DNA. We used helicase and SSB proteins to open up the two strands of DNA so we can expose all the nucleotides of the template. Where does DNA helicase unzip DNA? We said at specific locations within the chromosome, there are what we call origins of replication. And a helicase will come over here, helicase will come over here, helicase will come over here. And in fact, I should be more specific, multiple helicases join at the origins of replication, and helicases can open the DNA in two directions. One helicase could open up in this direction. Another helicase can open up in this direction. So we're just looking at the purple for now. So the purple bubbles are called replication bubbles. There are no hydrogen bonds connecting these the two purple strands anymore because DNA helicase came in and started unzipping. And now again, pay attention to each replication bubble. There are two replication forks. So what this means is that DNA will continue to be separated until we have two daughter molecules. What's being shown in blue is DNA polymerase making new DNA. So pause right here and try to get a sense of what this picture is telling you. Right, we start off with one chromosome with 2 strands of DNA. At multiple locations along that chromosome, DNA helicase unzips it and continues to unzip in both directions until the entire chromosome is unzipped and new DNA can be made from both template strands. A video might be helpful for you to watch at this point before moving forward. So now let's see how DNA helicase creates replication forks. Here we have a double-stranded DNA molecule, 5 to 3, 3 to 5. Great. Here's one of those origins of replication. So what's going to happen is DNA helicase, the enzyme, will come in, will recognize that origin of replication, and will start breaking the hydrogen bonds that join the 2 strands. So we separated the hydrogen bonds now. And DNA helicase will continue to go in this direction, breaking all of these bonds. So DNA helicase is going in this direction. Here's DNA polymerase. Now, where will DNA polymerase want to go? Right? Remember, DNA polymerase joins nucleotides 5' to 3'. So how could it do that? Well, if it binds over here, if DNA polymerase binds over here, let's say that's the DNA polymerase, then it can easily make DNA 5' to 3'. However, DNA polymerase can't attach here because this is the 5' end. And we can never have DNA being made 5' to 3' on top of a 5' strand. No, we can never have that happen. So there's going to be an issue here. DNA polymerase has no problem going to the 3' end and making DNA continuously. But we'll see there's another problem when we want to copy the other strand. So DNA has 2 strands. There's a leading strand that is built continuously, and then we have a lagging strand that is built discontinuously. Let me explain this step by step. We said DNA helicase unzips the DNA at one strand, unzips the DNA, and expose a 3' end and a 5' end. Look at only the light blue right now. Look at the light blue. That's the parent DNA molecule. DNA polymerase has no problem recognizing the 3' end here and then making new DNA from 5' to 3'. It can do this continuously because it's going in the same direction as helicase. So, and you're being shown an arrow, right? Helicase is unzipping this way. So DNA polymerase could basically follow the same path as DNA helicase and continue to make DNA 5' to 3' continuously without a problem. Great. So the leading strand, this is called the leading strand because it's made continuously in the 5' to 3' direction. And it's built continuously because DNA polymerase is going in the same direction as DNA helicase. The lagging strand, this strand over here, cannot be synthesized continuously because DNA polymerase must wait for the helicase to unzip the template ahead of it. This is a very visual process. It might not make sense right away, so let's try to break it down. We know DNA polymerase could not start here and just make DNA 5' to 3', right? It's impossible. What has to happen is it has to wait. So this is the first little fragment put down. The lagging strand is built from short fragments called Okazaki fragments. Okazaki is the scientist who discovered these fragments. So the leading strand is built discontinuously in short Okazaki fragments. So what has to happen is the second DNA unzips a little bit, then DNA polymerase can put down a short fragment 5' to 3'. And then has to wait for helicase to unzip more, and then DNA polymerase can come in and make a new fragment 5' to 3'. Then helicase will unzip a little more, and then we'll make a new fragment 5' to 3'. Right now it has a problem, right? How is it going to copy this DNA over here? It has to wait for helicase to unzip more, and then DNA polymerase can make a new Okazaki fragment there. So one strand is built discontinuously because it has to wait for helicase to unzip so it can make DNA fragments 5' to 3'. So I hope that makes sense at least a little bit. Right? One strand is made continuously, the other strand is made discontinuously. So I want to try to go over really quickly just a schematic of DNA replication. So up here in red, I have my parental DNA double helix, this ready to be copied. So suppose helicase will unzip over here, and we'll have a 3' end and a 5' end open. Where can DNA polymerase start? Well, it can't go this way, 5' to 3', because then it'll have a 5' here, and that will be parallel. We cannot do that. So DNA polymerase will start here toward the 3' end, 5', and will start following DNA helicase, 5' to 3'. Right now, at this point, DNA polymerase cannot make anything in this direction, 5' to 3'. It can only make one little fragment, 5' to 3'. So now the next step that has to happen is then DNA helicase will have to come in and then break these hydrogen bonds. And then what can happen after is then DNA polymerase can put down another fragment, 5' to 3'. Right? And then DNA helicase will have to break these bonds again. I'm not going to show that. And then DNA polymerase will put down another fragment, 5' to 3'. So that's showing how it's always going, it's lagging. It has to wait for DNA helicase to unzip in order for polymerase to put a little fragment down. However, with this top strand, DNA can be made in one go, 5' to 3', in one continuous strand. I hope that makes sense a little bit. So how do those fragments get joined together? Well, a special enzyme called DNA ligase is like DNA glue, and it joins all the Okazaki fragments together, sealing them into a continuous strand. Interestingly, DNA polymerase cannot just start replicating DNA on its own. It can only recognize another strand. So DNA polymerase can only add nucleotides to an existing strand. Therefore, it requires a primer in order to start. This little primer is a short sequence of RNA nucleotides shown in green here. So there's a couple of A, U, C's, and G's that are put down by a special enzyme called Primase. So Primase is the enzyme that kicks off DNA replication by making this little RNA primer. Then DNA polymerase can recognize that and start making fragments, or start making DNA. In this direction, it will be one continuous strand, but in this case, right, only short fragments. This was the first fragment to be put down, then another fragment. And for each fragment, RNA primase had to have come in. So primase is an enzyme that came in and put down a little primer of RNA, and then DNA polymerase made this fragment. Primase put a little fragment of RNA primer, and then DNA polymerase finished this fragment. The RNA nucleotides in the primer are later replaced with DNA nucleotides. So your chromosomes are 100% DNA. There's no more RNA left. Right? Because of lagging strand synthesis, the very ends of chromosomes do not get replicated. The ends of chromosomes are called telomeres. Telo always means end. And because of this lagging strand synthesis, it can never, it's hard to explain, but the tips of chromosomes can never be copied because helicase can't go ahead of the ends of the chromosomes and unzip it. So basically the ends of chromosomes just remain unreplicated, which means every time the cell divides, the ends of chromosomes, the telomeres, get shorter and shorter. So this is really, really important. So the shorter the telomeres get, the older the cell's age gets. So telomeres shorten as the cell ages. And when telomeres get to a certain length where they're so short, the cell dies. Telomeres are copied by the enzyme telomerase in certain types of cells, like stem cells and cancer cells. And that's what makes these cells immortal. So with the enzyme telomerase active, you can continually add ends to the chromosomes, and this way the cells won't die. And we'll talk more about telomerase when we talk about cancer. So let's review all the enzymes of DNA replication. This is one picture. I'll show you this next one. This whole thing takes about 8 to 10 hours, so it's very, very efficient. And then I'll go back to the slide after. So to summarize, we know replication requires helicase to unzip the parental DNA molecule. SSB proteins then attach to the separate strands to prevent the helix from reforming. In order to make new DNA, we need primase to first put short primers on the strands to be replicated. DNA polymerase will then recognize the RNA and begin DNA synthesis. The two strands are replicated differently. We know leading strands and lagging strands have different rates. Each daughter strand at the end will have one old and one new strand, and that's what makes DNA replication semi-conservative. I just want to go back and talking about the accuracy of replication. So DNA polymerase is very active, but it makes a mistake about 1 every 100,000 base pairs. This is okay though, because DNA polymerase also has a proofreading ability. So it can identify and correct the errors. And at the end, after proofreading, DNA polymerase has an error rate of only about 1 in a billion nucleotides. There are other repair enzymes that help the accuracy of DNA replication, that they can cut out any mismatch and replace with the correct bases, called the nucleotide excision repair. What would happen if DNA polymerase couldn't proofread? Right? There would be a lot of mutations, maybe too many mutations for our own good. But what would happen if polymerase never made a mistake? Right? Then there would be no genetic diversity, and there would be very little choice for nature to select from. So it's good that we make some mistakes every now and then, so we get different varieties, right? Different things for nature to choose from. So, what unzips the DNA double helix? Helicase. What copies DNA? Polymerase. What attaches DNA fragments together? Ligase. What makes the RNA primer? Primase. And here again, you can see in blue, DNA helicase started up here on the blue parental strand, but it went here and then it went this way. And since this 3' end is exposed, DNA polymerase, starting green, started here and then just built continuously the leading strand 5' to 3', following the direction of helicase. This lagging strand wasn't as lucky because it had to wait for helicase to unzip a little bit, and then this one fragment was put down. Then helicase unzipped a little more, and then this fragment was put down. Then helicase unzipped, and then this fragment was put down. Once this unzipped this way, one more fragment will be put down. And then this one, and then this one. And that's why it's kind of in reverse. That's why it's lagging strand synthesis. It's almost like backstitching. You have to keep on going backwards. So now's a good time to watch the animation included in the ebook that will talk about all the different enzymes in DNA replication. And here are some videos I'd like you to watch as well. So this is a cool video that shows DNA replication in real time. I'd like you to watch the whole thing in its entirety, but you can see here the DNA's unzipping. And I'll let the narrator explain what's going on specifically.
And copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact
That's
copy.
helicase.
So DNA comes in and hits this
Unzipping.
blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple as the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring the right way inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you.
So that's the end of DNA replication, and now we're ready to talk about the cell cycle. So the cell cycle describes the process of cell growth and cell division. And in cell division, we see a parental cell divides into two daughter cells. And we know that it had to replicate the genome first. Bacteria, we said, are so easy. They just replicate their genome by DNA polymerase, and they do what's called binary fission. And that is basically just splitting into two. The cells elongate, cytoplasm splits into two. That's binary fission. We have two identical cells. That's all you need to know about prokaryotes. They have a circular DNA chromosome, gets replicated. So you have two circles of DNA. The circles go to opposite ends of the cell, and the cytoplasm splits. Fission is separation, binary is two. So it's splitting into two. In eukaryotes, the cell cycle is a continuous process with many distinct phases. Each of your body cells right now is in one of these phases. And it's a cycle, so it continues to go on and on and on. Some cells can exit the cell cycle in what's called the G0 phase. We'll talk about that in a bit. But for the most part, all of your cells are in some aspect, some phase of the cell cycle. So in eukaryotes, we have two types of cells. Right? So there are two different types of cells in multicellular eukaryotic organisms. Our somatic cells, or our body cells, contain two sets of chromosomes, right? One set from mom, one set from dad. We call these diploid, right? Di for two. They have two sets of chromosomes, two copies of every chromosome. Specifically, we have 46 chromosomes. We have two copies of each of the 23. So in humans, N equals 23. That's how many chromosomes we have. And in a diploid cell, we have two copies of each of the 23. We have two N. So in an average somatic cell, say your hair cell, your skin cell, your eye cells, they have 46 chromosomes. They're diploid somatic cells. The other type of cell in multicellular eukaryotes are germ cells, or gametes, like sperm and egg. So gametes, like sperm and egg, only have one set of chromosomes. So gametes, like sperm and egg, have one set of chromosomes and are therefore called haploid, 1N. Sperm and egg are the only cells in the body that are haploid, and humans. So gametes are haploid. That means each sperm and egg cell has 23 chromosomes. So when a sperm and an egg come together, then we'll get a 46 chromosome set. So here you can see that gamete cells found in the testes of males or the ovaries of females are haploid. The rest of the body cells are diploid. Somatic cells are body cells. So somatic cell division is what requires mitosis and cytokinesis. And that's what we'll talk about today. So somatic cells divide by mitosis and cytokinesis. Every time you scrape your knee and need to repair your skin cells, this is mitosis. As a new baby is developing, that's mitosis. Right? Genital growth from child to adult is mitosis. And cytokinesis, I should say. So mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm. Together, mitosis and cytokinesis equals cell division. Mitosis gives us genetically identical cells. So that's an important distinction. So mitosis gives us genetically identical somatic cells. Right? Only somatic cells multiply by mitosis. Gametes are formed by meiosis. That's a very important distinction. So gametes multiply by meiosis. I should say they're made by meiosis. Whereas mitosis is found in somatic cells. So make sure that's clear in your notes. Today we're going to talk about somatic cells, their body cells, doing mitosis. And the next lecture we'll talk about how germ cells make gametes by meiosis. Somatic cells that are not dividing are an interphase of the cell cycle. So interphase is most of the cell's life. So most of your cells are in interphase. And just to go through this life cycle, sperm and egg are formed by meiosis. We'll talk about that next time. But after fertilization, we have what's called a zygote. It's the fertilized egg. That's the first diploid cell because it has 2 sets of chromosomes. And from that point on, mitosis makes cells with identical DNA, and that's what allows development to occur. And from birth until adulthood, it's a bunch of mitosis and development. Only after puberty does meiosis begin. So to review, we said mitotic division results in genetically identical daughter cells. The cell's endowment of genetic information is called its genome, and we said that cells must duplicate their genome before they divide to make sure that each daughter cell receives an exact copy. Cells must also duplicate all the organelles and enzymes before division, so each daughter cell has enough organelles and enzymes. In summary, if the cellular environment is just right, the timing is right, everything is good, the 2 DNA copies are allocated to opposite sides of the cell, opposite poles of the cell, and then a signal is sent for it to split down the middle into 2 daughter cells. That's what we're going to see in summary. Right, we started off with a parent cell, and then DNA polymerase has to come in interphase before mitosis begins and replicate all of those chromosomes. Once DNA is replicated, then we can divide into 2 daughter cells. And these 2 daughter cells will be identical to the parent cell. Remember that normally the genome is coiled as chromatin. And chromatin is DNA and proteins combined. When genes are active, we said DNA is loosely condensed as euchromatin. So RNA polymerase can access those promoters and make RNA from them. After DNA replication, we don't want any genes to be expressed. We actually want them to shut down. We want to just divide the cells. So after DNA replication, chromatin is condensed into heterochromatin to form visible chromosomes. In most animations and pictures in a textbook, you always see chromosomes drawn like this. But that's not really accurate. Chromosomes only look like this right before mitosis. Right? We have 46 chromosomes, 23 from mom, 23 from dad. All of them will condense before cell division. And this is just a repeat. We know each chromosome is a DNA double helix wrapped around histone proteins. They're, again, condensed. And once the chromosome is duplicated in interphase, each identical half is referred to as a sister chromatid. So these are identical sister chromatids. Each chromatid has a centromere in the middle. That's the middle of the chromosome. And in a replicated chromosome, we have 2 centromeres. Right? So telomeres are at the end. And again, chromosomes only appear this way during prophase of mitosis after duplication. The telomeres are at the end. The centromeres are in the middle. Kinetochores are special proteins that are attached at the centromere. And these will be very important when we want to pull these 2 sister chromatids apart. So an overview of chromosomes in mitosis. I'm just showing you one over here, but we have 46 of these in a non-dividing cell. 46 chromosomes. Each chromosome gets replicated during interphase by DNA polymerase. Then we get these. This is still considered one chromosome, but it's duplicated. So it has 2 sister chromatids, but they're attached together at the centromeres. So sister chromatids are identical copies of a chromosome that are held together at the centromere. There are 2 kinetochore proteins. And during a point in cell division, these 2 sister chromatids will be pulled apart. Remember, sister chromatids are 2 identical double helices. This will happen 46 different times. Well, I should say, right, 46 of these pairs will be separated. So at the end, we're going to have 46 chromatids in this cell, 46 chromatids in this cell. how do those chromatids travel to separate ends of the cell? Well, we need microtubules to help form what's called the mitotic spindle. So the mitotic spindle controls chromosome movements during mitosis. Can you recall where in the cell do microtubules come from? So in an animal cell, right, the centrosomes are the microtubule organizing center. Right? So centrosomes are structures in animal cells that can organize the mitotic spindle. Right? So we have all of these microtubule proteins that can then coordinate the movements of the chromosomes during mitosis. Each centrosome has 2 pairs of centrioles that can generate the microtubules. Plant cells don't have centrosomes. They organize their spindle fibers throughout the cell. So we won't really talk about that too much. You can pause here for some confusing terms up until this point. So I know centromere, centrosome, centrioles, chromatid, chromatin, all these sound a little confusing. Now's a good time to make sure you understand the terminology before moving forward. So it's possible to photograph cells as they're dividing using a technique called immunofluorescence. And this is a picture that I actually took myself about 5 or 6 years ago when I was working in pharmaceuticals. These are lung cancer cells in the process of dividing. And I was able to label 3 different macromolecules, different colors. So I'm going to bring this picture back at the end. I want you to guess what is labeled, what did I stain blue in all of these cells? What did I stain green in all of these cells? And what did I stain red? The red is the hardest to figure out. But try to pause here and come up with a guess. What is stained in red? What is stained in green? What is stained in blue? This is, again, called immunofluorescence, and I'll get back to this after. So for now, let's go talk about mitosis and cell division. So somatic cell division consists of interphase. Interphase is where the cells are going to grow, the DNA will be copied, and the cell is just prepping. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. So these are the 3 steps to somatic cell division, to the somatic cell cycle. Let's start with interphase. Interphase is most of the cell's life, right? And it's divided into 3 parts. There's first G1, the first gap phase. We have S phase for synthesis. That's when DNA, polymerase, will make DNA. Right? That's a very important step. And then G2 phase is the second gap phase. So starting from G1, again, the cell is going to grow, carry out its basic functions, produce new organelles as required. Then, if timing is right, the DNA looks good, then the cell will enter S phase of interphase, which is DNA synthesis. That's where the enzymes, DNA polymerase, will replicate the cell's genetic material and repair any damaged DNA. So in the beginning of S phase, each chromosome consists of 1 DNA molecule, but by the end of S phase, each chromosome consists of 2 attached sister chromatids. In G2 phase, the cell continues to grow but also prepares to divide, producing any protein needed to help with mitosis. The DNA will wind very tightly around its proteins and form condensed chromosomes, and then we're ready to enter mitosis. There's also what's called G0 phase, and that's cell cycle arrest. And in that phase, the cell continues to function, but does not replicate its DNA and does not enter mitosis. So nerve cells in the brain are permanently in G0. And that's why the brain does not grow after it reaches adult size. And that's why brain damage is often irreparable, because the cells don't do mitosis anymore. They can never be replaced. We'll see how important cell division is later on. So we'll talk more about interphase in a bit, but let's watch this video first.
The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap 1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis.
So, mitosis is divided into phases to help us kind of regulate what's going on. Again, it's a continuous process. It's not like there's a stopping point in the cell, but we're going to divide them up into phases. Prophase, prometaphase, metaphase, anaphase, and telophase. Prometaphase is not always included, but the book includes prometaphase, so therefore it is going to be on the quizzes. Cytokinesis starts during late telophase, and that's when the cytoplasm divides and we have two new cells. M phase refers to mitosis and cytokinesis together. So let's talk about prophase and prometaphase. During prophase, the DNA, the chromatin will condense into chromosomes. Those become visible. At the same time, the centrosomes move apart. Centrosomes move apart. So they start going to opposite ends of the cell. In prometaphase, which occurs immediately after the centrosomes start moving apart, the centrosome will start making the mitotic spindle made from microtubules. And the nuclear envelope breaks down. So far, prophase, we have DNA condenses into chromosomes. The centrosomes move apart. The nuclear membrane breaks down. And the spindle fiber starts to form. That's prophase and prometaphase. In metaphase, all of the chromatin pairs meet in the middle of the cell, and the microtubules attach to the kinetochore proteins at the centromere. So remember, there are two kinetochore proteins. The mitotic spindle attaches one to one each, so one on this end, the other on this end. And it's very important for all the chromosomes to line up along the equator of the cell, so they're divided equally. So if we zoomed in, here's two centromeres. These are identical sister chromatids, and the microtubules grab onto the kinetochore proteins at the centromere of each chromatid. So metaphase, chromosomes line up in the middle, microtubules attach to the kinetochore proteins, and the arrangement along the equator is very important. So we assure each cell will receive just one copy of each chromosome. So pause here and answer the following question. So the answer is B, right? B does not occur in prophase or prometaphase, rather in metaphase. Next up, we have anaphase. And in anaphase, the centromeres split, and the two sister chromatids are pulled apart. So each sister chromatid is pulled to an opposite pole of the cell. You can see this is a nice image here where the chromosomes are in blue and they're being separated. Here again, you can see this is a light microscope image, and this is in a plant cell. It's an animal cell and a plant cell. I should have specified that before. These are animal cells and these are plant cells. So anaphase, sister chromatids get pulled apart. You can remember A apart for anaphase. Right. So we've had prophase, prometaphase, anaphase. The next phase is telophase. So the easiest way to think about telophase is as the reverse of prophase and prometaphase. So whatever happened in prophase and prometaphase, the opposite will happen in telophase. Specifically, the nuclear membranes will be rebuilt around the two sets of chromatids. So we're reforming two new nuclei. Think about telophase as two new nuclei. The chromosomes will then relax and unravel back into chromatin. And the microtubules will disappear. The spindle will disappear. So in prophase, we said the spindle formed. In prophase and prometaphase, the spindle formed, the nucleus broke down, and the chromosomes condensed. Now we're doing the opposite. The chromosomes are decondensing, the nuclei are reforming, and the spindle breaks down. There's also the formation of what's called the cleavage furrow in animal cells. It's shown here, and it's a shallow groove that cinches the middle of the cell. And that's going to start cytokinesis. Right? Cytokinesis is division of the cytoplasm, and it occurs by cleavage in animal cells. What's microfilaments of actin, if you remember, actin is another cytoskeletal protein, form a contractile ring. And as this ring cinches, kind of pinches off, the cleavage furrow is showing this is the cleavage. The contractile ring is made of actin, and kind of like a purse string, cinches these. So the two cells are divided, and cytokinesis is complete. In plant cells, different vesicles are distributed along the middle, and what's called a cell plate is then formed between the two daughter cells, and that is then generated into a cell wall. So cytokinesis is different in plant cells than in animal cells. This is a good time to pause and recap each of the steps in animal cells and in plant cells. So what phase is being shown here? This must be metaphase. What phase is this? This must be anaphase.
Differential interference contrast microscopy is used here to visualize mitotic events in a lung cell grown in tissue culture. Individual chromosomes become visible as the replicated chromatin starts to condense. The two chromatids in each chromosome remain paired as the chromosomes become aligned on the metaphase plate. The chromatids then separate and get pulled by the mitotic spindle into the two nascent daughter cells. The chromatin decondenses as the two new nuclei form, and cytokinesis continues to constrict the remaining cytoplasmic bridge until the two daughter cells become separated.
Great. So now I'd like you to watch this animation very, very, very closely. This is also on the online resources folder. This is the best video I found that can summarize all the cell cycle at once. So please watch this as many times as you need.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing two copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has four stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
So now that we've spoken about cell life, let's talk about a little more of the death part. So we need to control cell life and death at all times. And a cell's longevity and lifespan, right, and the frequency of cell division will vary on a lot of different factors. So it depends on the cell's health, the cell's type, and what's going on around the cell, like the microenvironment. And regardless of what's going on inside and out of the cell cycle is under very tight control. So how does a cell know when to divide or not? Right? It's a very complicated question. And in cell biology, you would spend many chapters focusing on just how the cell knows when to divide. But in short, there are cell signaling pathways that tell the cell when to divide or not to divide. Specifically, we mentioned growth factors are special signaling molecules that can bind to receptors on the plasma membrane and cause some change on the inside of the cell to turn on the cell cycle. So very briefly, and in general, what will happen is a special growth factor will bind to a receptor. The growth factor can't cross the membrane on its own, so it needs to bind to a receptor first. And when it binds to the receptor, there will be a change on the inside of the receptor that can activate other proteins in the cytoplasm. These activated proteins in the cytoplasm can then send a transcription factor into the nucleus. And what we know about transcription factors is it tells RNA polymerase, hey, RNA polymerase, transcribe this gene. So as a result of this transcription factor being there, RNA polymerase will then make this growth gene. We'll start transcribing the growth gene. And then a protein will be made that can stimulate cell growth and mitosis. So again, in response to this growth factor, the nucleus changed the gene expression to make proteins to stimulate mitosis. Inside of the cell, these proteins called cyclins go up and down. They go up and down. They cycle, and they trigger various phases of the cell cycle. So they increase and decrease over the course of cell division. And again, the concentration always fluctuates in very predictable ways during each stage. So for example, cyclin E always peaks between G1 and S phase. Cyclin B always peaks between G2 and M. And what happens is these cyclins pair up with other special proteins called cyclin-dependent kinases. And these kinases activate the different phases of mitosis. And that's where I'm going to leave it at that for now. But these cyclins always fluctuate, and those pair up with these special enzymes called cyclin-dependent kinases that are dependent on cyclins. And these can then activate different parts of mitosis. So the cell cycle has specific checkpoints where the cycle stops just by default. So this is like you can't just hop on an airplane if you want, right? You have multiple checkpoints that you have to go through in order to get on the plane. The checkpoints can only be overcome if the proper go signals are received. These signals will ultimately either activate or inhibit enzymes of cell division and control whether cyclins are produced or destroyed. And again, that's a little more detail, but right now I want you to understand that there are these built-in checkpoints and think about why that's important. Cell division should only be done when it's necessary, right? We don't want cells to divide unless it's 100% required and the cells are healthy to begin with. So checkpoints keep the cell cycle under tight control. Checkpoints ensure that all the DNA has been replicated properly, DNA is not damaged, the chromosomes lined up properly and separate properly. Specifically, these are the main checkpoints. Before a cell even replicates its DNA, there's the G1 checkpoint. Is the DNA damaged? Do I even want to copy my DNA? Then, if so, DNA looks good. Okay, I'll go on with S phase. But then there's another checkpoint. Did all the DNA get copied? If not, it will not be allowed to proceed. So at this point, before it enters mitosis, has all the DNA been replicated? Can the damaged DNA be repaired? Is all of my spindle machinery in place? If yes, then we can start mitosis. And within mitosis, there's a checkpoint at metaphase that asks, do all kinetochores attach to the mitotic spindle? And are the chromosomes aligned down the equator of the cell? That's very important. If not all the chromosomes are attached to the microtubules, then there won't be equal division of the chromosomes. For example, I will draw something as best I can. So suppose I have, that's one chromosome, and here's another chromosome. So suppose in this organism, there's only 2 chromosomes. And microtubules pull from each side. So if there are 2 microtubules, then these are going to be separated, right? This will be pulled apart into one, and then the other, like that. However, let's say microtubules are only attached on one end, and there's no microtubule here. Well, then these 2 chromatids will go to one cell, and nothing will go to the other cell. So that's why this metaphase anaphase checkpoint is very important. We want to make sure all of the microtubules are attached to the kinetochore proteins. So if these are not perfectly in favor for cell division, then checkpoint arrest happens, right? We stop the cell cycle. Anytime you see this kind of arrow, that means inhibition. It's an inhibitory arrow. So if there's an unfavorable environment or damaged DNA, we stop, we arrest in G1. If there's incompletely replicated DNA or any damaged incompletely replicated DNA, then we stop in S or G2. If the chromosomes are improperly attached to the mitotic spindle, we stop at the metaphase anaphase transition. So this is, again, keeping everything in check and making sure our cells divide with fidelity and they don't make any mistakes. So you can pause here to review. Something I should note is that if there is DNA damage, the cell might repair the damage and then try again. But that cell can also kill itself if it's not necessary, if it's not repairable. I should also mention that at this point, if cells are not destined to go onto S phase, they could just stay in G0, which is that senescent phase of just hanging out and not dividing. So let's review this quick animation together.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
So we spoke about the regulation of cell growth and division. There has to be a balance, right, between all of that and cell death in order to maintain homeostasis. So as cells divide and survive, we need some cells to die. So apoptosis, that's how we pronounce it. Apoptosis is programmed cell death or cell suicide. And this is the only form of suicide that is actually very good, because our cells are doing this to protect the rest of the organism. So apoptosis is programmed cell death, and it's a very tightly controlled process. Necrosis is another type of cell death that's ugly. They just kind of burst open, and that's really bad cell death. Apoptosis is programmed, so it's actually very tightly controlled. For example, in embryonic development, tadpoles need to become frogs. So all the tail cells have to undergo apoptosis. They program suicide. At a certain point in development, they just all kill themselves. So therefore, the tadpole does not have a tail anymore and can become a frog. When we, or like for birds or amphibians, they start off with webbed feet. And for humans, for example, we need to sculpt our digits from that webbing. So the webbing gets killed by apoptosis. Here's an example of where apoptosis failed, right? There should have been killing of these cells, but that did not happen. And this is called polydactyly, when you have digits fused together. Our neurons always undergo apoptosis as they develop. As our brains develop, neurons try to find their right pathways, and any neuron that doesn't find the right pathway has to kill itself. In healthy animals, our bone marrow always undergoes apoptosis because we want new bone marrow cells, right? We want new immune cells, so we want to kill the old ones to constantly replenish. Same idea in our intestines. Our intestinal cells are constantly being generated, so we need to kill the old ones. And those are done on a cycle. They're programmed to be killed in an elegant way. Most importantly, and what we'll talk about for the rest of the time, is apoptosis happens in genetically mutated cells. If there is a cell with genetic mutations, with DNA damage, that cell will kill itself. Without that, we wouldn't be here today, right? So when there is DNA damage in a cell that cannot be repaired, the cell can kill itself by apoptosis. Apoptosis is triggered by enzymes called caspases. And what caspases do is they cleave all the macromolecules into fragments. So they chop up all the DNA, all the proteins, all the fats, until the plasma membrane blisters into what are called blebs, little fragments. So mitosis and apoptosis are opposing forces. Mitosis increases cell numbers, apoptosis decreases cell numbers.
Under the microscope, we can see how cells are destroyed. This process continues throughout our lives as cells become damaged or just worn out.
So a critical point in the cell cycle is the G1 checkpoint. After things are, the cell's growing, this is the critical point where it has to say, am I supposed to divide? Or am I supposed to pause? Or am I supposed to die? Right? So a functional G1 checkpoint is critical to maintain that balance between cell life and death. Because at this point, if it sees a go signal, it will continue on to S phase to replicate its DNA. If there is a stop sign, if the cell does not receive a go signal at the G1 checkpoint, the cell might go into a non-dividing state like G0, or it might go to apoptosis. So at the G1 checkpoint, the cell has to take into considerations the internal health of the cell, all the signaling inside of the cell, but also what's going on the outside of the cell. What growth hormones are present? What is the environment like? So if the environment is good, the DNA is intact and not mutated, the cell is healthy, then it will likely go on to divide. What happens when things become imbalanced? Well, unfortunately, many diseases that affect humans are a result of the cell cycle being imbalanced. In cases where we have too much cell death and not enough cell division, growth, and survival, we have neurodegenerative diseases like Alzheimer's and Parkinson's. Remember, neurons are non-dividing. So if only we can understand the cell cycle a little better and get them to divide or understand why they stop dividing, maybe we can understand Alzheimer's and Parkinson's better. On the other hand, suppose we have way too much cell division, growth, and survival and not enough cell death. Well, that's when we get cancer. And that is what we'll talk about for the remainder of this lecture. So, to recap, we said cancer results when things become imbalanced, when there's too much cell division, growth, and survival, and not enough apoptosis and cell death. Then things become imbalanced, and cell growth becomes uncontrolled. So what cancer is characterized by uncontrolled cell growth and spread of abnormal cells that may result in cell masses called tumors. The key word here is uncontrolled. Cancer is when cell division gets out of control. So that's the biggest distinction. Over here, you can see in this liver, there are multiple growths. These are tumors that have metastasized, meaning they spread from an initial site in the liver. This might have originally come from skin. This could have come from like a regular skin cell. But because of the process called metastasis, as we'll soon learn about, it can go to other organs of the body and colonize. Over here, you can see that tumor cells stain differently than normal cells. And here you can see these tumor cells are trying to invade these normal cells over here. So there's first a myth, right? A lot of people assume that cancer is inherited, that cancer is a genetic disease. And cancer is more of an age-related disease than anything else. This graph over here pretty much shows that as you increase in age, the incidence of cancer skyrockets, right? So if it were a genetic disease, we would see mostly children being affected, right, if their parents had it. But that's not the case. Cancer is an age-related disease. Some cancers are heritable. So there are cancers like that are congenital we see in children that are due to a specific mutation. But for the most part, cancer is an age-related disease that's due to uncontrolled cell growth. And we'll talk about why those cells lose their control over division. So what causes cancer? Genetic mutations lead to cancer. Right? We said mutations are alterations in the genome's genetic instructions. A mutation is like a typo in DNA. So DNA typos may lead to cancer. We'll talk about that not any typo will cause cancer. Right? But what can mutate? What can even cause these typos? Chemicals called carcinogens or mutagens can alter genomic DNA. And cigarettes are good examples of substances that are full of carcinogens. There are 250 harmful chemicals in tobacco smoke. 69 of them are proven to cause cancer. And what's interesting is this is showing the per capita cigarette consumption peaked in the 60s, in the mid-60s. And then you see the death rates from lung cancer and how that followed about 30 years. So it's mutating DNA over time, right, with age, can give you cancer. What else can mutate DNA? Well, UV light, even from the sun, or radiation can physically break apart DNA strands. Certain viruses can insert their DNA into the infected host genome. So for example, HPV, the human papillomavirus, can insert itself into certain cervical cells. Hepatitis virus can insert itself into liver cells, and this can damage our DNA. It can interrupt the normal message, the normal instructions that our DNA has. But again, it's not just, oh, anytime I mutate DNA, you get cancer. It's specific types of genes that, when mutated, can cause cancer. Let's go back a little bit. We said that the cell cycle is regulated by a control system with checkpoints. And the cell cycle is continuous but has specific checkpoints where the cycle arrests until a go signal is received. It turns out that two types of genes control these cell signals for proper cell division. It is very important to write these two things down. Proto-oncogenes are types of genes that promote the healthy cell growth and cell division. So proto-oncogenes are pro-division. They cause cell growth and division in a healthy way. Growth factors are example of proto-oncogenes. So growth factors promote cell growth and division. We like proto-oncogenes. We need them. The other type of genes are called tumor suppressor genes. And those do the opposite of proto-oncogenes. Those inhibit cell growth and division. So tumor suppressor genes are ones that are going to cause apoptosis and say, stop dividing. It's not your time to divide. So these two genes are critical. Proto-oncogenes and tumor suppressor genes work together to make sure there's the proper homeostasis and the proper control over the cell cycle. So this is where we get into trouble. We said proto-oncogenes are good genes that promote the cell cycle. But if I mutate a proto-oncogene by UV light, by chemicals, cigarettes, anything, a proto-oncogene then becomes an oncogene. Onco is a prefix for cancer, right? Oncology is the study of cancer. An oncogene is what leads to uncontrolled cell growth and division. So we start off in a healthy cell with a nice proto-oncogene that's telling our cell to divide normally. Something, whether it's UV light or a chemical, mutated this proto-oncogene into an oncogene. And now this is telling the cell to grow uncontrollably. And that can give us a cancerous cell. The other family of genes, those tumor suppressor genes, are the checkpoint genes that stop the cell cycle unless it's required. Examples are called P53 and RB, a retinoblastoma gene. P53 is one you must know about. We'll talk about that next. So a tumor suppressor gene normally tells the cell to stop dividing, right? Normal tumor suppressor gene makes a protein that inhibits growth, so it keeps cell division under control. However, if I mutate my tumor suppressor gene, there's no more stop signals to tell the cells to stop dividing, so cell division will not be under control. P53 is the classic tumor suppressor gene. It is called the guardian of the genome, and it's that important. P53 is activated by DNA damage. This is a really cool phenomenon. Active P53 can then stop the cell cycle and initiate DNA repair. So normally, P53 detects any DNA damage, it gets activated by DNA damage, and then it'll tell the cell cycle to stop. It will give the cell a chance to repair its DNA. If not, P53 will say, kill yourself. They'll say, do apoptosis, you're not good for the organism, you should die. P53 is so important, right? Because P53 can cause apoptosis if the DNA cannot be repaired, and therefore will prevent cancer. So another way to think about how clever this is, suppose you're typing a report, and the first time you make a typo, then spell check is activated, and everything is double checked and make sure that it's good. It's the same idea. It's like the first time there's DNA damage, then everything is checked, and if it's not good, it's not going to be allowed to divide. You can imagine what happens if you have a mutant P53 then. If you have a mutant P53, even if you have DNA damage, the cell cycle won't stop. The DNA will not be repaired, and the cell will continue to replicate with that damaged DNA. And it sure, makes sense because more than 50% of all tumors have a mutation in P53. So this just shows how important P53 is if more than half of all tumors have a P53 mutation. So I'd like to give you an analogy to understand this a little better. So suppose, like when we drive a car, all cars undergo everyday wear and tear, right? We get dents, we bump into things, there's some scratches, paint chips, and that just happens as cars get older. right? But still, as they get older, it's still usually safe to drive. However, there are two types of damage that can lead to a fatal car accident that, regardless of how old the car is, it's going to, crash. Suppose the brakes don't work, right? Your brakes don't work. That means you're going to drive right off a cliff. You're going to keep on going, nothing to tell you to stop. Similarly, if the ignition is always on and can't be turned off, you're going to drive off of that same cliff. So in this example, the brakes are like tumor suppressor genes that tell the car to stop or to tell the cell to stop dividing. Ignition is like the proto-oncogenes that normally tells the cell to go or the car to drive. But suppose my ignition is always stuck on. That's like a mutant oncogene. That's telling the cell to go, like the car's going, and it's going to drive off the cliff. So again, just to re-illustrate, right now your body is being bombarded by UV light. There are some chemicals in the environment, I'm sure, that can damage DNA. But most genes can be damaged and just be repaired. Or those cells can be killed by apoptosis. It's only when tumor suppressor genes or proto-oncogenes are mutated where the cell can't take control over the cell cycle. And that's when we get cancer. So it's a very important distinction to understand. Cancer is caused by genetic damage to proto-oncogenes or tumor suppressor genes, thus making the cell cycle uncontrolled. So cancer risk can vary by ethnicity because there's one such tumor suppressor gene called BRCA1. And certain ethnic groups have a mutation in BRCA1 that is heritable. So since BRCA1 is inherited, it makes it more likely for certain ethnic groups to get breast cancer. So BRCA1 normally prevents us from getting breast cancer. If there's a BRCA1 mutation, right, just like a p53 mutation, that means that there's no more tumor suppressor gene to tell the cell to stop dividing if there's a mutation. So a large proportion of Ashkenazi Jews have a BRCA1 mutation on chromosome 17, which makes breast cancer, BRCA1 positive breast cancer, more prevalent among Ashkenazi Jews and Hispanic populations. So there is a genetic component to cancer, but cancer is not just inherited. Right? Just because your mother has breast cancer does not mean that you have breast cancer. It might be more likely to develop breast cancer, but there's no direct causation. So reducing DNA damage reduces cancer risk, right? We said inherited mutations can't be really fixed without gene therapies, but factors in the environment can be avoided, right? So avoiding cigarettes, avoiding tanning beds and excess sun tanning, eating healthy diets, getting regular exercise, having safe sex can all protect you from cancer risks. Which of the following is not a cancer-causing gene? So please pause here. And the answer is C. A proto-oncogene is not a cancer-causing gene. It's a healthy gene that tells our cells to divide properly. Which of the following may damage DNA? So please pause here. And the answer is D. All of the above may damage DNA. Hopefully, after we suntan, right, all those red cells are going to undergo apoptosis. Right? Sunburn is basically telling your cells to kill themselves. And that's why your cells peel off. Right? So why is cancer so deadly? So what if we have some cells growing? Well, it's when the cancer cells spread through our tissue and go through the bloodstream and form tumors in other parts of the body that gets deadly. And this is called metastasis. Right? Stasis is in place, but now it's going out of place. So cancer cells first grow and grow and grow in situ, in place, but then they can invade surrounding tissues by getting inside the lymph and blood vessels. And once it gets inside to the circulatory system can transport those cancer cells to other parts of the body, and those cancer cells can then reinvade and grow new tumors. That's why cancer is so deadly. Why skin cancer, right? Skin cancer doesn't sound that bad. Oh, it's skin. Well, what started off as a melanoma, which is a skin tumor, traveled through the bloodstream to the liver and the brain. And when you have a brain tumor or a liver tumor, that's a lot more deadly, but it's still called melanoma. right? We call cancers based on where the tumor formed initially. So again, skin cancer could be very, very deadly. Not all tumors are cancerous though, right? Benign tumors are not metastatic. They stay in place. They do not invade. They're usually encapsulated, so they don't spread. A malignant tumor can invade adjacent tissue, and it does not have a surrounding capsule. It is most likely to metastasize. So it's going to go through the bloodstream or the lymphatic system to colonize other parts of the body. And again, it's over time. So one cell might have one mutation, but then that allows it to grow a lot. If there's an accumulation of more mutations, it'll allow it to grow and grow and grow until it has the advantage of being able to metastasize. So this is called the multiple hit hypothesis. Tumors accumulate multiple mutations as they spread. So it might start off as a benign tumor that has a capsule of connective tissue that keeps the tumor from spreading, might accumulate more and more mutations. And what happens is as those tumors acquire mutations, they can lose their capsule and have the ability to seed new tumors throughout the body by going through blood vessels or lymph vessels. Right? So that's where it gets metastatic. And tumors are heterogeneous, meaning within one tumor, each cell might have different mutations. And this gives cancer many different advantages and the abilities for them to divide and invade our cells. So the more mutations and the more diverse the mutations, the harder it is for our body to fight against cancer. And this increased genomic instability allows for even more mutations. So more DNA gets deleted and amplified, and cancer cells just become a mess of the genome. Tumors also alter the metabolism, so they use sugars differently than our healthy cells do. One way that tumors can do metastasis is via angiogenesis. Angiogenesis is how tumors form their own blood vessels. Angio means blood vessel. Genesis is formation of. So angiogenesis helps nourish the tumor with more blood supply, which could overall allow the tumor to grow and spread. Tumors are very smart. They release a chemical signal called VEGF, which is a growth factor. And that growth factor tells our own blood vessels to go inside of the tumor. So our blood vessels are tricked into growing inside of the tumor so that the tumor has an increased blood supply. This goes both ways. Not only does the tumor get more oxygen from our blood supply, but the tumors have a direction, a little shortcut into our circulatory system. Without angiogenesis, cancer cells die due to lack of nutrients. So it's no surprise that there are some drugs being formed that inhibit angiogenesis. So in this image, you can look at it I'm not going to talk about it too much at all, but mice and humans with cancer were treated with endostatin. Does it work in mice? Does it work in humans? So I'd like you to pause here and tell me what you think. So it seems that endostatin was administered to mice, and as it was administered, the tumors shrunk. But once endostatin was not administered, the tumors grew back again. But then when it was administered, it grew back down. So it goes up and down and up and down. Every time the endostatin was administered, they showed a decrease in the tumor size. And overall, at the end, after several days of treatment, like over 100 months of treatment, they saw a final reduction in tumor size. It wasn't as effective in humans, but it was still doing something. So with the chemotherapy, it seems like, without the control and untreated, the tumor pretty much grew in 30 days. With the chemotherapy drug, that tumor did not grow to the same size until twice as long, until after 70 days. So the endostatin helped delay tumor growth in humans. It was a lot more effective in mice. Drugs like Avastin, available now, can block activation of the VEGF receptors and inhibit angiogenesis. So now VEGF inhibitors like Avastin can be used in certain types of cancers in humans, and that blocks the VEGF receptor from causing angiogenesis. I want to talk about how different cancer cells are from healthy cells. So they're morphologically, meaning shape and structure, but also functionally distinct. Cancer cells lose contact inhibition. Normal cells have what's called contact inhibition, meaning they stop growing once they contact each other. So they never grow on top of each other. Cancer cells don't care. They'll grow right on top of each other. They don't stop growing even when they contact each other. Right? They can even form 3D clusters of cells. Telomerase, we said, is overexpressed in cancer cells. In normal cells, telomeres gradually shorten as cells divide. And after about 50 divisions or so, cells don't have telomeres and they stop dividing. In cancer cells, they have the enzyme telomerase that always extends their telomeres. So there's always going to be long telomeres and the cells will not stop dividing. That makes them almost immortal. Right? Telomerase constantly extends telomeres, making cancer cells immortal. That brings us to this very important slide that talks about the 6 hallmarks of cancer. And all these hallmarks kind of sound the same, or not sound the same, but they sound redundant. But it's important to understand each and every one of these hallmarks. All cancer cells resist apoptosis, right? They don't undergo cell death. They continue to grow and proliferate. Right? They sustain proliferative signaling, meaning they're always told to grow. And they evade growth suppressors. So any stop signs, they evade. They don't listen to any stop signs. Cancer cells can induce angiogenesis, formation of blood vessels. They can activate invasion and metastasis, right? They can spread. And they also have replicative immortality because of that telomerase. So they can divide as many times as they want. So this is a review. I'm not going to read this to you, but this is a basic summary of the differences between cancer cells and healthy cells. So what is not a hallmark of cancer cells? Pause here. So the answer is C. Cancer cells do not have reduced rates of mitosis. They have increased rates of mitosis. So this is an overview of causes of cancer. We said that growth factors normally tell the cells to divide, but if they're mutated into an oncogene, then it's going to be uncontrolled cell division. Tumor suppressors normally inhibit the cell cycle, but if you mutate a tumor suppressor, then you're going to have nothing to tell the cell to stop dividing, and that can cause cancer. So I'd like you to watch this animation that's included in the lecture folder that has all the online activities. Just talks about what is cancer. And here's the link down here. So just to save some time, I'd like you to watch that on your own. And here's just a reference for you. You're not going to be tested on this, but these are different types of cancer. So you might hear a sarcoma is a solid tumor of bone and cartilage. Carcinoma arises from the epithelial lining. So epithelium is like skin, for example. A lymphoma is cancer of lymph tissues and immune cells. And leukemias are cancers of stem cells that produce white blood cells. Now that we spoke about cancer and why it's so bad, let's give some good news, right? So when a person is diagnosed with cancer, we have a variety of weapons to combat it. Radiation uses ionizing energy to specifically target and kill a tumor by breaking its DNA. So again, we said that radiation could damage our DNA, but we could use very high levels of radiation that are specifically targeted to cancer cells. So we're going to only kill the cancer cell's DNA. Surgery, of course, can physically remove the tumor and surrounding tissues. That can be a little more invasive, though, since all of the surrounding tissue has to be removed as well, not just the tumor. Chemotherapy drugs are used to kill cancer cells by disrupting some aspect of cell division. And what are some side effects of chemotherapy? So you can pause here and think about it. And I'm sure what comes to mind is hair loss as one, and also nausea. And can you think about why people lose their hair and get nauseous from chemotherapy? Well, it's because chemotherapy is toxic to healthy cells too. Chemotherapy is just a chemical that kills cells. And it kills cells that rapidly divide first. So of course chemotherapies will affect cancer cells that are rapidly dividing. But they're also going to affect your hair cells, which are rapidly dividing. And they're also going to affect the cells of your intestine, which are constantly dividing. So that's why you lose your hair and we get nauseous when we take chemotherapy. What structure can be targeted to disrupt cell division? So what structure in the cell can we target by a chemical to stop the cell from dividing? Well, microtubules, right? Remember, microtubules pull chromosomes apart. If microtubules cannot pull chromosomes apart, then the cell can't divide. And it turns out a drug called Taxol was a chemical found in the bark of a tree, the yew tree. And it turns out that Taxol can bind to microtubules and stop them from moving. So microtubules are made of tubulin, and scientists figured out the structure of tubulin, and it shows where this Taxol binds. And when it binds to tubulin, it cannot grow or shrink. So with Taxol treatment, microtubules cannot pull chromosomes apart. Where do you think Taxol would arrest the cell cycle at? The answer is D. So at metaphase, anaphase, all the microtubules are kind of frozen into place when they're treated with Taxol, and they cannot go on to anaphase. So let's watch this brief video on how cancer could be treated.
Every year, it is estimated that there are over 12 million new cases of cancer worldwide. The type of therapy that a doctor will recommend depends on the type of cancer, how far it has progressed, and where in the body it is located. But the majority of cancer treatments fall into one of three categories: chemotherapy, radiation, and surgery. Chemotherapy kills or slows the growth of cancer cells within the patient's body using drugs. It is a more likely choice when cancer is located in several parts of the body. Radiation also kills or damages cancerous cells. Particular types of focused energy, called ionizing radiation, are directed at the site of the tumor. The DNA of the cancer cells is damaged, preventing them from replicating. This may slow the growth of the tumor, stop it, shrink the tumor, or kill cancer cells that remain after surgery. Various methods are used to direct the radiation at only the cancer cells, minimizing damage to nearby healthy tissue. In the case of surgery, the intent is to remove the tumor rather than to kill the cancerous cells that make it up. The potential benefit and risks of surgery will depend greatly on the type of tumor, its location, and a number of other factors. Because it involves cutting into the body, surgery almost always entails some side effects, although some newer minimally invasive techniques, such as laparoscopic and robotic surgery, can help to reduce them.
So as we continue to learn more about cancer biology, we'll be able to develop more cancer therapeutics. And we're heading in a direction toward personalized medicine. So it's going to be less of a one-size-fits-all. Oh, you have breast cancer, you take this chemotherapy. But we can actually do high-throughput genetic screens for patients. And there might be a possibility of taking a biopsy of a tumor or some kind of growth, and you can look at all the different mutations that could lead to cancer. And based on the specific mutations of that tumor, we can have data saying, oh, with this mutation, then it's best to take chemotherapy X at a low dose for 7 days. And then if you have this mutation, then you should do a different chemotherapy. Right? So as we accumulate more data, and as we develop more DNA sequencing technology, I predict that we're going to see a lot more high throughput genetic screens that can pinpoint types of mutations and then can direct the oncologist what kind of medication to administer. There are also some really cool therapies being worked on right now that can reprogram the cancer patient's immune cells to recognize and destroy the tumors. This is called CAR T therapy. And you could take out, if somebody has cancer, you can take out their immune cells and reprogram them to recognize the cancer as being foreign. And you can put those immune cells back into the patient, and then the patient's own immune cells will take care of the cancer. That's, the best way to go possibly, because right now you have cancer cells in your body that your immune system is just detecting and they're killing them. Our immune system normally makes sure that we don't have any cancer in our body. So with CAR T, you could basically reprogram a patient's immune system to do what it's supposed to do. One final technology that's being worked on, or not one final, so there's many, many, many things. One final one I'll mention is the use of viruses that only kill cancer cells. So this is a really interesting thing. So you could give a virus, you can infect the patient with a virus, but this virus only attacks cancer cells because they don't have p53. It's a long story to explain how this works, but the nutshell is you're specifically targeting only cancer cells that don't have p53, meaning those are the cells that can't correct their DNA damage. All the other cells that have p53, which can correct DNA damage, are fine. To put it all together, we said mitotic division has three stages: interphase, mitosis, and cytokinesis. DNA replication by DNA polymerase occurs in S phase of interphase. Mitosis is division of the nucleus, right, with chromosomes, and that occurs in phases: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis is division of the cytoplasm that occurs during late telophase. And finally, we said if mitotic cell division is uncontrolled, it leads to cancer. So the following slides are not going to be tested on, but they're interesting and important for you to know. These are estimated new cases of cancer in 2020, with prostate cancer leading male cases, breast cancer leading female cases, and in both males and females, lung cancer is the second most popular. In both male and female, lung cancer is the most deadly. So about 25% of all cancer deaths are lung cancer deaths. And this is an interesting graph that looks at new cases on the x-axis and the mortality of cancer on the y-axis. So over here we have the most common cancers, and the higher we get is the most aggressive, most deadly cancers. So lung cancer is something that is very common and very aggressive. This explains why a lot of pharma companies are trying to figure out how to cure lung cancer, how to treat lung cancer. In every project I've been on in cancer research, working in pharma, it was a lung cancer drug. On the other end of the spectrum, we have these very rare, very deadly diseases like esophageal cancer, pancreatic cancer, and liver cancer. Those are more risky to try to cure. So pharmaceutical companies might be interested in coming up with a cure to treat these, but it's very, very hard to do. So we still have a ways to go. The good news is that mortality is decreasing over time in the general trend. The incidence, again, is overall kind of stabilizing. Females have a higher prevalence of breast cancer and lung cancer slightly, but overall mortality is going down. Right? Mortality is going down, which is a good thing. If you're interested in learning more, I highly recommend the book, The Emperor of All Maladies, which is basically a biography of cancer. And it talks about how cancer was discovered and some of the earliest cancer patients and what they had to go through. And finally really talks about the war against cancer. And it's also a documentary that you could watch as well. Another book I highly suggest is The Immortal Life of Henrietta Lacks. And Henrietta Lacks was a poor African-American woman living in Baltimore in the 50s, and she went to Johns Hopkins because she had a cervical tumor. And the doctors at Hopkins took a sample of her tumor, and to their surprise, those cells just kept on growing. They just grow and grow and grow. They didn't ask for her permission. So this was a bioethical issue. That was, again, against her will. Her family didn't even know about it. But scientists held onto her cells and took them into space to study the effect of gravity on cells. They injected them into animals. They did all these genetic manipulations. In fact, Henrietta Lacks' cells are still alive today. And if you were to work in a cell biology lab, you would use HeLa cells. We call them HeLa cells after Henrietta Lacks. So I've used HeLa cells many, 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 many times because they just grow so well. So in a way, this woman, Henrietta Lacks, is still immortal because her cells are being used by scientists around the world. And I'd like you to watch that video talking a little bit more about Henrietta Lacks. And here finally are some division terms that get a little complicated. You must know all these terms before moving forward to study for the quiz. Lastly, I promised you I would show you this picture again, and you can see these round nuclei in blue, right? That means I stained the DNA blue. Right, so DNA is stained blue, all these nuclei. Here we can see a dividing cell. So these chromosomes are being separated to opposite ends of the cell. So what's in green? What are pulling the DNA? Microtubules. So I stained microtubules in green. In a non-dividing cell, microtubules just form the train tracks, right, of the cytoskeleton. But in a dividing cell, they form the mitotic spindle, and they can pull chromosomes apart. Finally, the red dots are the centrosomes that form the microtubules. So you can see two centrosomes form the microtubules and are pulling those chromosomes apart to opposite ends of the cell. So with that, we finished chapter 8. Thank you for listening. And next time we will talk about meiosis and sexual reproduction.